Hello everyone, this presentation will instruct you on how to prepare a cheek cell slide. In the previous experiment, you prepared an onion cell slide and you looked at a plant cell. Today, the cheek cell slide is an animal cell. So once we have the data, we'll be comparing both plant and animal cells. Once again, we're using a stain, so gloves on the hands and eye goggles. Methylene blue is a common lab stain. It's used for many different types of cells. And in the case of the human cheek cell, methylene blue causes the DNA in the nucleus to stand out so that the nucleus can clearly be seen in a light microscope. What's really interesting about methylene blue as an indicator is that it can tell us whether the cells are dead or alive. The stain cannot penetrate live cells. However, in dead cells, it does penetrate the cell membrane. And so the stain blue will clearly show the cells. Step 1. Place 1 drop of methylene blue on the middle of the slide. Step 2. Take a clean cotton swab and gently scrape the inside of your mouth. Now I want you to scrape along the inside of the cheeks, back and forth, up and down, inside of the cheeks, not the tongue, the cheeks. And you will be getting a lot of saliva on the cotton swab, along with cheek cells. Step 3, you'll roll the cotton swab in the methylene blue drop for about 4 to 5 seconds. Just roll it around and the cells will drop off the methylene blue. Don't worry if you don't see anything, they're there. Just as a word of caution, don't put the cotton swab back in your mouth. Thank you. Step 4, gently lower the cover slip. Remember that 45-degree angle from yesterday. Use a paper towel to carefully dab away any liquid that leaks out. Step 5, place the slide on the stage. Looking at the diagram above again, the slide goes onto the stage and is held in place with clips. Don't clip a slide in place until you have found an area to look at. If you happen to look inside the eyepiece and you don't see any cells, unclip the slide clips, move the slide, refocus, find those cells, and then clip the slide back onto the stage using the stage clips. Read for further instructions in the packet carefully. Turn to page 3. Once again, you will be listing both qualitative and quantitative observations under descriptions, and be sure to be very accurate with that drawing. Help yourself to the colored pencils if necessary. Once again, a reminder about our cleanup. Please clean the equipment. All equipment needs to be properly washed, rinsed, and put in the drying area. Put away all the equipment. Put back all equipment to where you originally got it if you do not have to wash it. Again, if there's any stain on the microscope itself, please wash it off. Put away goggles neatly into the storage area, which is our black crate. Be sure to store the microscope with a revolving nose piece turned back to the low power objective lens and lower that body tube. I'll send you back if those microscopes aren't being stored properly. Rinse off all slides and cover slips, and please put them in the solution, and once I feel that they're adequately washed, I'll put them on the paper towel pad to air dry. Please let me know if any are cracked or broken. Wipe down the table if needed. Stay in your seat at your station until dismissal. I noticed there was a little bit too much traffic yesterday. So once you take care of your role, you get your job done, go right back down to your seats so that people can move around safely. This presentation has been made for my spectacular students at JMS. Please feel free to review this anytime you need. Thank you.